Good morning, everyone. I'm delighted to welcome you all today. Today, I will be presenting on a very important topic. Dr. S.K. Singh, Ph.D. Routine chemical and microbiological analysis workflow. In this session, we'll explore the key steps involved in routine analysis processes, and I'll share some insights to help streamline and improve your workflows. Let's get started. Today, I'll be discussing the main objective of our procedures, which is to establish standardized methods for handling production samples in the QC laboratory. These procedures are critical because they ensure data integrity, compliance with regulations, traceability, and that the samples remain unaltered throughout the process. We want to make sure that every step, from receiving the samples from production to their final disposition, is consistent and reliable. The scope of these procedures covers every stage, starting with the receipt of samples, then moving on to proper registration and labeling to avoid mix-ups. Next, we focus on storage and the controlled distribution of samples to the analysts, which helps maintain their integrity. When it comes to analyses, whether chemical or microbiological, following standardized procedures is essential for obtaining accurate and reproducible results. After testing, the data review and report issuance are crucial steps to ensure the results are valid, complete, and compliant. Finally, we address the proper disposal or archival of samples, which is important for traceability and future reference. These procedures apply to all types of samples, raw materials, in-process materials, and finished products, covering the entire life cycle from initial receipt to final disposition. Overall, this comprehensive approach helps us maintain high-quality standards, regulatory compliance, and confidence in our testing processes, which ultimately supports the integrity of our products. Let's start by looking at some key definitions that are fundamental to our understanding of quality control and testing in the pharmaceutical industry. First, a sample refers to a portion of a batch that is specifically released for quality control testing. This is the material we analyze to ensure it meets all the required standards before it reaches the market. Next, the chain of custody is crucial. It's a documented transfer record that tracks the sample's journey from collection through testing. This documentation ensures traceability and integrity throughout the process. Then, we have the analytical method, which is a validated procedure used for testing our samples. Validation here means that the method has been thoroughly tested to be reliable, accurate, and repeatable. Holding time is another important concept. It's the maximum allowable time between sample collection and analysis. If we exceed this time, the sample's integrity could be compromised, affecting our results. Now, it's essential to recognize the key references that underpin these definitions and practices. These include good manufacturing practices, or GMP, as outlined by WHO, US FDA, and EMA, which set the standards for quality and safety. We also look at ICH-Q2, R1, which covers analytical validation, ensuring our testing methods are robust. The principles of GLP, or good laboratory practice, guide the quality and reliability of our laboratory work. ISO 17025 is another critical standard, focusing on testing competence and laboratory accreditation. Lastly, various pharmacopoeial standards like USB, IP, and BP provide official quality benchmarks for pharmaceuticals worldwide. All these references help us maintain consistency, reliability, and compliance across our testing processes, which ultimately supports the overall goal of ensuring safe and effective medicines. Let's take a closer look at this sample receipt from production. First, we want to verify the document itself. That means checking the sample transfer form or batch records to ensure everything is properly documented. We also need to confirm the number of containers and the sample type matches what was expected. These steps help us ensure traceability and accuracy right from the start. Next, we move on to the integrity check. It's crucial to verify that the seal is intact, which indicates the sample hasn't been tampered with during transit. We also want to make sure the quantity is correct. Additionally, inspecting the conditions of transport is essential. Any signs of damage or exposure that could affect the sample's stability or integrity. These verification steps are vital to maintaining quality and confidence in our samples, aligning perfectly with our overall goal of ensuring product integrity from production to analysis. Now, let's talk about labeling and storage. Proper labeling is crucial to ensure that each sample can be easily identified and tracked throughout the testing process. The label must include the sample ID, which uniquely identifies the sample, as well as the batch or lot number to trace its production details. It's also important to specify the test type, whether it's chemical or microbial, so everyone knows what analysis is being performed. Additionally, including the date and the person who received the sample helps maintain accountability and traceability. When it comes to storage conditions, maintaining controlled temperature and humidity is essential to preserve the sample's integrity. Also, chemical and microbial samples should be stored separately to prevent cross-contamination and ensure safety. Moving on to sample registration, the first step is to assign a unique sample ID. This can be done through an electronic system or a manual register entry. All relevant details, like the batch number, product description, quantity, date and time of receipt, and storage conditions, must be captured accurately. It's equally important to record who handles the sample, specifically the QC staff responsible for receipt. This creates a clear chain of custody and ensures accountability at every step. By following these labeling and storage protocols, we uphold the quality and reliability of our testing process, which is central to the overall integrity of our work. Let's walk through the microbiological analysis workflow. First, we start with media preparation and a sterility check to ensure our growth media are free of contaminants, which is crucial for reliable results. Next, we move to inoculation and incubation, where we introduce our samples into the media and allow them to grow under controlled conditions. After incubation, we perform enumeration. This includes total plate count, yeast and mold counts, and pathogen detection, to assess microbial presence and levels. If applicable, we also conduct environmental monitoring to verify that our surrounding environment remains clean and contamination-free. Throughout this process, accurate data recording is essential, and we incorporate controls to validate our results. Switching gears to chemical analysis, we begin with method selection and verification, ensuring our testing methods are suitable and reliable. Reagent preparation and standardization follow, which guarantees consistency in our assays. Instrument calibration and qualification are vital steps to maintain accuracy and precision. Sample preparation involves weighing and diluting samples properly to prepare them for testing. Then, we perform various tests, such as assays for active ingredients, impurity analysis, and dissolution testing. All raw data is meticulously recorded to maintain traceability. Once data collection is complete, samples are distributed to analysts in a controlled manner. This includes maintaining a distribution log with timestamps, noting the analyst's name and signature, and providing clear instructions regarding priority and expected completion dates. Preservation requirements are also critical. Some samples require special storage conditions like refrigeration or light protection to prevent any alteration or degradation. Altogether, these steps form a comprehensive and rigorous workflow that underpins the integrity and reliability of our testing processes. Now, let's look at what the report includes. First, it shows the test results compared to the specifications, so we can quickly see if everything passed or if there are any issues. It also clearly states the acceptance or rejection status, which helps us understand whether the product meets the required standards. Additionally, you'll notice there are signatures from the analyst and reviewer, ensuring accountability and traceability. Finally, the report is dated, which is important for documentation and record keeping. Moving on to data review and report preparation, the key principles are that the review should be complete, accurate, and traceable. This means every piece of data must be checked thoroughly, and we need to be able to follow the data back to its source. We review raw data, calculations, and visual evidence like chromatograms or images. By adhering to these principles, we ensure the integrity and reliability of the report, which is essential for quality assurance and regulatory compliance. Now, let's talk about the release decision. Once the quality assurance team has completed their review, they will evaluate the report carefully. They have the authority to approve or reject the batch release based on the findings. If there are any out-of-specification results, they will document the justification for proceeding or holding the batch. In some cases, corrective and preventive actions, or CAPA, may be necessary to address any issues identified. It's also important to note that all of these decisions and actions are thoroughly documented to maintain a complete audit trail. This process ensures we stay compliant with regulatory standards and quality requirements, which is crucial for maintaining the integrity of our products and trust with our customers. Now, let's talk about sample disposal or archiving after analysis. It's important to follow the retention policy, which typically ranges from 3 to 5 years. This ensures we keep samples long enough for any future reference or review. When it's time to dispose of samples, we need to do so in a controlled manner, especially for hazardous materials like chemical waste or microbiological samples. Proper disposal methods are crucial for safety and compliance. Additionally, maintaining disposal records is a key part of good laboratory practice. These records should include the date of disposal, the method used, and the authorization for disposal. This documentation helps us stay compliant with regulations and provides a clear audit trail. Overall, proper disposal and archival practices are essential components of responsible sample management and align with our commitment to safety and regulatory adherence. Now, let's talk about some best practices for ensuring quality and compliance in our processes. First, it's crucial to always follow validated methods. This means using procedures that have been tested and approved to give us reliable results. Next, we need to make sure that our instruments are properly calibrated and that these calibrations are up to date. This helps us maintain accuracy in everything we do. It's also important to document any deviations from our standard procedures and the corrective actions we take. This documentation creates a clear record and helps us learn from any issues that arise. Maintaining a chain of custody is another key point. It ensures that samples or materials are tracked securely and responsibly throughout the process. Lastly, periodic training and audits are essential. Regular training keeps everyone up to speed on best practices, and audits help us verify that we're consistently following them. All of these practices together support our overall goal of maintaining high quality and compliance standards in our work. For further support, please feel free to contact us. We're here to help with any questions or additional information you might need. And finally, I want to thank you all for your time and attention today. It's been a pleasure sharing this information with you, and I hope you found it valuable. Remember, we're always available to assist you as you move forward. Thank you.